All right, so let me just go back to Unit 7. So we started out Unit 7, we started talking about cellular respiration. We talked about how it had different stages. There was glycolysis, which we talked about, and from there we go to pyruvate oxidation. And if I'm right, I think we left off before getting into Krebs cycle, or maybe we didn't even get into pyruvate oxidation. Maybe we just finished glycolysis, I think. So why don't we just pick up at the end of glycosylation. So when we look at stage 1, which is glycolysis, at the end of glycolysis, at the end of these 10 steps that make up glycolysis, we are left with 2 molecules of pyruvate, and we have made 2 molecules of ATP. We have made 2 molecules of NADH. So then what happens? So then these molecules of pyruvate can now enter into the mitochondria to enter the next stage of cellular respiration, which is dehydrogenation by pyruvate dehydrogenase. So this pyruvate molecule does not contain very much energy. And so through the action of coenzyme A, it can be transformed into a molecule called acetyl-CoA by the attachment of coenzyme A. A molecule of CO2 is released, and it started out with a 3-carbon molecule of pyruvate, and acetyl-CoA is now a 2-carbon molecule of a 2-carbon molecule. Excuse me, I am I've got a really terrible cold. I'm so sorry if I'm sniffling for everybody. So then this 2-carbon molecule of acetyl-CoA enters into the Krebs cycle. Now, overall, if you look at Krebs cycle, and again, it's a cycle, so it regenerates. So this Krebs cycle, and another name for it is the citric acid cycle, its ultimate goal is to rearrange the carbon molecules that form in order to remove electrons along the way and store the energy in these molecules of NADH. And because we have, if we look back at our pyruvate oxidation state, we see that from each molecule of pyruvate, we get 1 molecule of acetyl-CoA. So if we look back, we see that these 2 molecules of acetyl-CoA are ultimately formed from 1 molecule of glucose. And so essentially this Krebs cycle has to happen twice for every molecule of glucose, because every molecule of glucose produces 2 acetyl-CoA, and each acetyl-CoA that enters and essentially goes through this, cycle has to happen twice, if you think about it, for each molecule of glucose. So here in my notes, I've tried to label each step for you so that you can follow along, but it's also identified very clearly here. So we start out with each acetyl-CoA molecule here, and this acetyl-CoA molecule then attaches itself to another 4-carbon molecule called oxaloacetate here. And this is at the very beginning, at the top of the cycle. So we have 2 carbons here from acetyl-CoA, we have 4 carbons from oxaloacetate, and we end up with a 6-carbon citrate molecule. And this citrate molecule rearranges itself to isocitrate. In the next step, isocitrate loses one of its carbons in the form of CO2, and the resulting 5-carbon molecule of alpha ketoglutarate then becomes oxidized. And through its oxidation to the molecule of succinyl-CoA and the release of a CO2 molecule, NAD plus becomes reduced to NADH. So here we have one CO2 released and another CO2 released during Krebs cycle. So if we keep going, then this succinyl-CoA becomes converted into the molecule succinate. And during this step, a molecule of ATP is formed by substrate level phosphorylation, and coenzyme A becomes released. So here you can see that the coenzyme A, this CoA that was attached here up until this point of the cycle, is now released, and this CoA now goes back to join to become used up again here back in this step. So then this succinate then, which has 4 carbons now at this point because it has lost 2 carbons along the way as CO2 in these 2 steps, this becomes converted to a molecule called fumarate. And during this process, 2 of its hydrogen atoms are used to reduce the molecule of FAD to FADH2. In the next step, fumarate is converted to malate, and then malate is converted back into oxaloacetate, again through the loss of hydrogen atoms to reduce NAD plus to NADH. So in total, we have glucose producing 2 pyruvate molecules in the cytoplasmic side, and then those pyruvate molecules enter the mitochondria, become converted to acetyl-CoA, and acetyl-CoA in the mitochondria then goes through Krebs cycle. So you can see from glucose to pyruvate to acetyl-CoA and 2 times around on the Krebs cycle. So finally, in the 3rd phase of cellular respiration, electrons become harvested from this oxidation of glucose that took place in the previous phases. These electrons then become transferred to the inner mitochondrial membrane. And this is the inner mitochondrial membrane. Here is the intermembrane space, and here is the matrix side. And these molecules of NADH and FADH2 carry electrons over and pass them along through a series of enzymes that act as electron acceptors and move these electrons through this electron transport chain that is embedded in the membrane. And as these electrons get passed down from the first electron acceptor, which is a protein called FMN, or flavin mononucleotide, short FMN, the protons that came along with these electrons also get transferred and end up in the inner membrane space. They become transported as a result of the energy that these 2 electrons bring with them. The protons that also came along become transported and go across into the intermembrane space. And these electrons then get passed down along the electron transfer chain from acceptor to acceptor. As they move along, again, these protons are able to use that energy and move into the inner mitochondrial space in a way that every electron pair is actually moving six protons from the matrix side into the inner membrane side. So finally, the concentration of the protons increases on the intermembrane space, creating a gradient across the membrane. A concentration gradient of protons and that electrostatic gradient of the potential energy, this is called the proton motive force, or the PMF, and the potential energy that's generated across the membrane that provides the energy for the formation of ATP, then is used as the protons move back across the membrane through a special channel. And this channel is the enzyme ATP synthase that becomes activated, and energy is released and is used by the enzyme to phosphorylate ADP into ATP. And this is called oxidative level phosphorylation. Let me see if I have a picture of it here. Yes, this is the enzyme ATP synthase. So as these protons that all collected across the inside the intermembrane space want to return back and travel down this proton gradient, the energy of this transport is used for oxidative phosphorylation of ADP to produce ATP. So by the end of the process, in total, one molecule of glucose produces in total 36 molecules of ATP. And if we want to look at the energy summary of that, we can look at the glucose that enters glycolysis. Glycolysis produces 2 NADHs, which get converted to 2 FADH+, and also within glycolysis itself, 2 ATP is made. For every 2 FADH2, you get a conversion to 4 ATPs. And then in the process of the oxidation of pyruvate to acetyl-CoA, you get 2 NADHs, and for every 2 NADHs, you can get 6 ATPs at this level. And then acetyl-CoA enters the Krebs cycle, producing, because there's 2 of them, 6 NADHs, 2 FADH2s, which ultimately produce 18 ATPs from the NADH and 4 ATPs from the FADH2 for a grand total of 36 ATP molecules. So this is in total just a brief summary of cellular respiration. There's also anaerobic pathways that are used to produce energy during times where there isn't enough oxygen available. Just 1 second, please. So if we look at cellular respiration, really the only phase out of the ones we looked at that uses oxygen, that doesn't use oxygen, is anaerobic, is glycolysis. So if we look back at glycolysis, during this stage, we see that it's anaerobic. There's no need for any oxygen. So when you're in an anaerobic state and you don't have oxygen present and oxygen is limited, this phase glycolysis is able to continue and keep on producing, at least for a period of time, a certain limited amount of energy. And the limited factor for this pathway is basically how much NAD plus you have available in order to reduce to NADH, because in order for glycolysis to go on, what you need to feed into the path is NAD plus. So as long as you're able to supply glycolysis with a supply of NAD plus, you can allow glycolysis to go on. So that is the limiting step for glycolysis, is the NAD plus, is one of the limiting steps. So if we go back to our notes, so as long as you have NAD plus available, it can be reduced to NADH. And if you have NADH that cannot be oxidized, glycolysis will eventually then shut down and no energy will be produced. And ultimately, this will mean death for the cell and eventually for the whole organism. So organisms have typically evolved two main pathways to allow glycolysis to continue when oxygen is available and also when oxygen is not available. So both of these methods, though, involve the transfer of hydrogen atoms from that NADH to other organic molecules in a process that's called fermentation. And we can take a look at one example of fermentation, which is ethanol fermentation. And this process is used by yeast cells, and also lactic acid fermentation, which is used by multicellular organisms, ourselves being an example of an organism that can make use of lactic acid fermentation. So let's compare and contrast these two ways. In ethanol fermentation, you have NADH, which transfers its hydrogen atoms to a molecule called acetaldehyde, which then leads to the formation of ethanol. So NADH2 passes on its hydrogen to acetaldehyde and gets converted to NAD plus, and as a result, you have this molecule of ethanol. And ethanol is exactly as you know it, alcohol that you have in alcoholic beverages. And when this happens, this transfer then again frees up NAD plus that can go back to glycolysis and keep glycolysis going to produce 2 ATPs for each run of glycolysis. Now for yeast cells, this energy that is made is enough to satisfy a small organism such as yeast's energy needs, but for us, we're not a small organism such as yeast. We use a different systems. But what we have done is we have taken advantage of the fact that yeast is able to carry out this process, and we use this alcohol fermentation that yeast carry out to make wine and beer and make ferment fruit juices and the sugar that is in them to produce wine, and that contains the ethanol. Also, we use this process during baking, where we use the CO2 that is released during the step where pyruvate is converted to acetaldehyde during the step in fermentation. And this bubbling of CO2 out from the reaction in the dough causes our dough to rise and leaven, while the alcohol that's produced will be burned off during the baking process. So that's what takes place in yeast. in multicellular organisms, we have lactic acid fermentation, and lactic acid fermentation happens in muscle cells, for example, for us when we don't have enough oxygen available to use for the aerobic portions of cellular respiration. And you have probably had this feeling of muscle fatigue, or sometimes if you've ran really hard, you might feel like your muscles are all cramped up and tightening, and after strenuous activity, you might feel that your muscles are in quite a bit of pain, and this is as a result of lactic acid fermentation. That clothespin activity that we talked about at the beginning of our lesson, during that portion, you can think about how as oxygen becomes less available because you just keep using it and you keep working this little muscle over and over, the NADH molecules that transferred their hydrogens to pyruvate, which then regenerated this NADH and allowed glycolysis to continue. This transfer converts pyruvate into lactate or lactic acid. And as lactic acid continues to build up in the muscles, our fingers become stiff or sore, or sometimes even our leg muscles, if we've been running, we feel very stiff and sore. That's because of the buildup of lactic acid. And then this pathway can be reversed again once oxygen is present, once we've rested and allowed oxygen to replenish again. So this lactic acid gets transported to our liver, where it can become oxidized back into pyruvate, which can then continue on through the rest of the pathways that require oxygen for cellular respiration and provide even more energy. But in the absence of oxygen, at least this little cycle here, this little step involving glycolysis and lactic acid fermentation can produce at least a small amount of ATP, a small amount of energy. So we've discussed photosynthesis and we've discussed cellular respiration. Let's take a look at the two and really compare and contrast these two processes and look and see how they are closely related, and look at the interdependency between these two pathways. So in the case of plants contain both chloroplast and mitochondria and can go through both processes. While animals, we don't have any chloroplast. We can only use up the energy that's provided by plant material, such as this glucose, to carry out cellular respiration. So essentially, the products of photosynthesis, especially glucose and oxygen, are the reactants that are necessary that feed into cellular respiration. While the products of cellular respiration, so if you look at cellular respiration, the products of cellular respiration, such as carbon dioxide and to some extent even water, these are products that are needed for photosynthesis. Both of these organelles possess some similar structures, which are used in similar ways, such as their interchondrial and inner membrane being folded to create more surface area, both in the chloroplast and in the mitochondria. And the presence of that electron transport chain that's embedded in that inner membrane and used to create ATP, and the presence of enzymes within that electron transport chain that are similar between the two and sometimes even exactly the same. And so there's quite a bit of similarities, but the important point to keep in mind is that both of these processes have developed over time as a result of evolution as a way to create the energy that cells need in order to perform all the essential functions that they need to carry out. So this diagram shows really the interdependence and how these two processes and these two organelles are actually quite interconnected and interdependent. How the product of one feeds into the other, while the product of the other feeds into this one. So that is the end of lesson 7 on cellular respiration. And we can carry on right into discussing cellular energy, into lesson 8. So really what we talked about over our last 3 lessons, we've been talking about energy transformations, how they apply to metabolic processes, namely photosynthesis and cellular respiration. And we have talked about the metabolic pathways that each one of these processes uses to convert energy into usable forms. And we've looked at these in detail. So overall, we should have at least a basic understanding how both plants and animals can either create, in the case of plants, or capture organic molecules that store chemical energy. So knowing this, we can look at the wider implications of how we try to control these processes. And there are definitely some benefits and some drawbacks as we try and control these processes. And one of the hot topics is really obesity and weight control that has become such a major issue in North American society. And as we all know, proper nutrition is very important to our overall health. And this is very closely tied to maintaining a healthy weight. And although there are many diets and many weight control supplements, some of these diets and some of these weight control supplements may actually do more harm than good. There's also weight control drugs that have been at some point approved for use that were then later proven to be actually dangerous and sometimes even deadly. And there's also been advertisements along the way for diet pills and miracle diets and with claims of miraculous results. So this really understandably creates a lot of confusion, but armed with our knowledge of metabolism, we can start to sort of weed through all of this confusion because we understand really all the detailed steps of cellular metabolism. Scientists have now turned to some plants or even some microbes, some bacteria, in the hopes of using some of their metabolic processes to also help to improve the environment. And some of these organisms, these microbes or some plants are capable of either absorbing or sometimes even metabolizing toxic waste in the material. So this is another way of using the metabolic processes within plants and microbes and really harnessing the utility of those transformation to look at how they could help to clean up environmental disasters such as oil spills, or to help to remove toxins, for example, the presence of cyanide from waterways and water sources. And so this has really opened up a whole new area of research called bioremediation. So that's going to be another topic that we can look at during this lesson. And some of the things you should keep in mind as we discuss the material in today's lesson is think about, do fad diets actually work? And what is a healthy way to lose weight? And should people use diet pills, and are they safe? And finally, on the topic of bioremediation, how can we use plants and other organisms to help to improve our environment? So first, let's take a look at nutrition and cellular energy. So although most people, at least on the surface, are aware that a balanced diet is important for good health, not everybody actually understands the role that the various nutrients actually play inside our body. And as we learned earlier, glucose, which is a carbohydrate molecule, is the main fuel source that our body uses to generate ATP. So if we have excess carbohydrates, they can be stored short term as glycogen in our muscles and livers. And then when our body needs additional energy, this glycogen that's stored in our muscles and liver cells can then be quickly converted back into glucose that enters the cellular respiration pathway and produces energy for us as we need it. And good dietary sources of carbohydrates are all the things you are typically probably familiar with: pastas, rice, whole grains, and breads. These all have carbohydrates in them. The more complex carbohydrates are the ones that are recommended over the simpler ones. That is because complex carbohydrates tend to have more complex molecular structures that take longer for our body to digest and break down. And this helps us to have that feeling of fullness and our meal sort of feeling like it's lasting longer without really needing us to go back to eat more. Also, these complex carbohydrates, in addition to the carbohydrates that they have, they also contain more vitamins and dietary fibers, which have been stripped away and are missing when carbohydrates become refined into sugar or white rice or white flour used to make white bread. So that's why complex carbohydrates are generally recommended over the simple refined carbohydrates. Fats is another source for energy. And fats for a long time have really been viewed as being bad for our health. And what you have to understand that it's not the fats themselves that are unhealthy, but the amount of fatty food that we eat in our North American lifestyles and North American diets, and the type of fat that we eat. Fats, also known as lipids, can be used to make ATP. That's another way that your body can generate ATP when carbohydrates are not readily available to the body. When fats are digested, they are broken down to give glycerols and fatty acids. If you can remember back when we looked at the structure of fats and lipids, we could see that they are made up of a glycerol molecules and 3 fatty acids. So the glycerol gets converted into that molecule of pGal, which then enters that cellular respiration path during glycolysis. So we can flip back to our glycolysis reaction pathway and see where pGal enters the way. So the glycerol from fats gets converted to pGal and enters glycolysis, and then the fatty acids get stored as fat, or they get converted to a 2-carbon fragment that acetyl-CoA, which can then, in lipid molecules, include such things that we can eat as butter or cream or margarine or various oils. Also, they are very much present as the fat that is found in meats and in nuts. And typically, you have plant oils generally recommended over animal fats as being healthier. So although fat may not be the body's first choice being the carbohydrate, fat also does get converted into producing energy. The other molecule that the body is also able to metabolize, it's not the body's first choice, but is protein. And proteins can also be broken down to create ATP. And protein is made up of various amino acids, and these various amino acids can get converted into substances that then enter the cellular respiration in many different places. We're not going to really go into this in very much detail, but it's important to understand that the protein molecule, this macromolecule that is found in protein sources such as red meat or poultry or fish, seafood, eggs, milk products, and even in plants, beans, peas, nuts, these are all good sources of protein, which can also be converted to produce energy. Also, you have cereals and pastas that to some extent contain protein. I mean, if you think about it, all the enzymes that carry out all the functions, all of those are made out of protein. So even if in a molecule, in a grain of cereal or in pasta where you have carbohydrates, there is also along with it some amount of protein. So the recommendation goes, excuse me a minute. All right, so it is recommended that approximately 65% or so of someone's daily energy needs should come from carbohydrates, with another 35% or so from fats or lipids, and then the remaining whatever small amount from proteins. So proteins, you only need a very small amount from. Now, according to Canada's food guide, and the food guide gets updated every once in a while, and it's good to take a look at the most recent version if you're really interested and want to know these numbers. Adults should make sure to have about 7 to 10 servings of vegetables and fruit, 6 to 8 servings of grain products, 2 servings of milk and alternatives, and about 2 to 3 servings of meat and alternatives in order to stay healthy. This will allow our body to meet its needs, not only for energy, but also for vitamins and minerals and other nutrients that will contribute to our overall good health. In this way, we also reduce the risk of obesity and type 2 diabetes, not to mention heart disease and certain types of cancer and osteoporosis. Now, what about these fad diets? So if you really talk to nutrition experts, it's always recommended that the safest and the most reliable way of losing weight and keeping it off is to change our overall lifestyle and to change our habits and lifestyle overall in a lifelong way. And to do this, we need to cut out about 500 calories a day and begin some sort of an exercise routine. But in many cases, in order to cut these excess calories means that we have to readjust what is our serving selections, or try to match our food intake to more closely match what's recommended in the food guide. And theoretically, this will allow our extra weight to come off gradually. But it's very tempting to give in to ads that we see on TV. After all, gradual weight loss is a very slow process. You have to look at it as a lifelong process. So it's very tempting to look at ads that we see in TV or magazines or on YouTube or social media that suggest that you can maybe lose 30 pounds in 30 days or lose weight while you sleep. And then you have various celebrities that swear by one diet or another. So many of these diets don't follow the guidelines, but rather very severely reduce or sometimes completely eliminate one of the food groups. And this, while initially can cause a person to lose weight in the short term, but this can be very unhealthy from a metabolic standpoint, because eliminating certain food groups altogether, together with that energy source that you're eliminating, you're also eliminating vitamins and minerals and other key nutrients. So one of the fad diets that you may be aware of is the low fat or no fat diet. So in this diet, people really severely limit the fats that they eat. And then the fats, what happens is they get replaced with, you guessed it, even more carbohydrates. The idea being that if you're not eating fats, your body can't make fats. And I hope you know that by having looked at cellular metabolism, this is completely untrue. You can have glucose and convert it into other molecules, such as glyceraldehyde 3-phosphate, which can then in turn be converted to glycerol. And glycerol, as you all remember, is part of the lipid molecule. It can also be converted into pyruvate, which can then be converted to acetyl-CoA, and then into fatty acids, which are really the other part of the lipid molecule. So here you have all you need in order to create lipid molecules. So really, any excess energy, no matter what nutrient it originally came from, if it's in excess, if that energy is in excess, if it's more than what you need, will get stored ultimately as fat. So with this diet, not only will you continue to create fat, because although you're not eating fat, you are replacing your dietary needs through your carbohydrates. Not only that, but this person's sugar levels will quickly rise because of all the extra carbohydrates that they are consuming. So this diet can be quite problematic. Now, how about the low carbohydrate, high protein diet? And this diet goes by a number of names. Sometimes it's got the author's names on it, but anyhow, no matter what it's called, the basic idea in this type of diet is that you can eat unlimited amounts of proteins, such as lots of meat, lots of poultry, fish, cheese, eggs, while allowing almost no carbohydrates, so low that essentially it's limited to negligible amounts. And this means eliminating such important foods as most vegetables are not allowed, certainly no sweets, rice, pasta, breads, fruits, and milk and dairy. So this really results in initially an immediate weight loss, but most of that weight loss is as a result of water. So when a person then goes back to slowly starting to eat carbohydrates, because it's almost impossible to continue to maintain this diet, that water weight is then regained. And if it so happens that the person is able to maintain this diet for a long time, eventually some muscle weight will be lost. And so if you look at this diet, there are many problems with diet. And anytime you have a diet that is not addressing all the food groups, you tend to worsen chronic and acute conditions and make it harder to recover from various illnesses. For one thing, the high protein in this diet and the high fats in this diet will increase the risk of heart disease, cancers, and kidney disease. And then the lack of carbohydrates will deplete all the muscle glycogen stores and can make exercising and activity seem much harder. So you feel a lot more lower in your energy. It can also lead to very unpleasant conditions such as nausea and diarrhea as a result of the lack of the fiber that comes in with consuming complex carbohydrates and vegetables, and also constipation and fatigue. Also on this type of diet, a great deal of calcium gets lost as a result of high protein levels. Because as a result of this diet, you typically don't get as much calcium as you need to take in because of the dietary restrictions. Calcium is oftentimes present in many vegetables and in dairy products such as milk. So calcium levels are often reduced in this sort of thing. But the biggest problem with this type of diet is a dangerous problem that is called ketosis. And during ketosis, you have ketones. And if you remember from your chemistry and from some of the chemistry that we covered earlier in our unit 1, ketones, and an example of one is acetone. So acetone tends to build up in the blood as a result of the incomplete breakdown of fats, these stored fats. And what this does, it gives very bad breath and actually can cause the pH of your blood to become quite acidic. And now blood pH is something that needs to be maintained, is very closely maintained through homeostasis. But if you, through this sort of a fad diet, this type of an extreme diet, and ketosis allow the pH of the blood to drop down and become very acidic, then this significant shift from what is blood's normal natural pH results in the denaturation of various proteins and enzymes, and denatured enzymes and proteins can't work properly anymore, and this will lead to eventual to cell death, and will ultimately affect the person as a whole. So I hope we can all see that really the recommended route of overall lifestyle shifts rather than these very severe fad diets, that is the way to go. The problem with these fad diets is that they tend to ultimately cause more harm than good. The recommendation is to gradually change your lifestyle, incorporate more exercise, and try and more closely follow the Canada Food Guide, consuming complex carbohydrates, and a good amount of leafy green vegetables and fruit. Small amount of protein, small amount of fat. So then, what about diet pills and medication? So these weight-reducing medications can really be divided into two main categories. And the two main categories are ones that you can get through by over the counter, and ones that you need a prescription for. So prescription drugs and over the counter drugs. And really, the over the counter drugs are the ones that tend to have very small amounts of whatever is the active substance that supposedly makes them safer. And there's also some natural or homeopathic products that contain ingredients that studies have suggested may have an effect on appetite. Again, there's oftentimes no conclusive evidence with these things, but regardless of the type of weight loss drugs, typically the mechanism of action is one of these three. They either suppress your appetite, or they increase the body's metabolism, or they somehow interfere with your body's ability to absorb some specific nutrient in the food. So the ones that suppress your appetite do exactly as you suspect or promise to do exactly that, to reduce your wanting, your cravings to eat. And oftentimes they contain amphetamine-like substances, and some of these have very serious side effects, including increased blood pressure, dry mouth, constipation, headache, insomnia. And there are some names of some drugs here as examples. The other category is weight loss drugs that increase your body's metabolism. So these drugs then increase the rate of your metabolism by stimulating your central nervous systems. And the problem with these is that these can be very addictive and habit forming. So these are not good either. And then the third type is ones that interfere with your body's ability to absorb specific nutrients in food. And some of these drugs block the breakdown of fats. And as such, because you don't break down your fats, you're preventing fat absorption. The problem is that your intestines have not evolved to be able to contain a lot of fat, have a lot of fat move through them. And when you block the breakdown of fats, what the side effect ends up being is that you have very oily bowel movements. And this can lead to quite a bit of cramps and stomach pain and flatulence. The problem with oily bowels is also leakage. So none of these are very pleasant side effects. Some of them are quite dangerous. So really, in general, not a good idea. It's really best to take the gradual approach and slowly try and reduce your caloric intake and increase your exercise and your level of activity, whatever that may be, to however amount. It doesn't have to be anything too extreme. So over the years, there's been actually some pretty bad press about some drugs. Fen-phen and redox is an example of one of them. And fen-phen was thought it was actually a combination of two drugs, phentermine and fenfluramine. And this was most commonly prescribed diet medication in the 1990s. So actually not too long ago. And then there was this drug redox, which was developed as an alternative to that fenfluramine and thought to have fewer side effects, but eventually both drugs got removed from the market in September 1997, where there was enough evidence to show that both of these drugs caused heart valve degeneration in up to 30% of those who had used it. So pretty significant numbers, and pretty dangerous drugs with pretty dangerous effect. So now we're going to switch tack, and we are going to talk a little bit about bioremediation. We started to talk about that in our introduction. Now we're going to get into it. So bioremediation is the process of using living organisms, such as plants or microbes, to clean up contaminated soil or water. And bioremediation can be broken down into three main types: biostimulation, bioaugmentation, and intrinsic bioremediation. So let's have a quick chat about what each one of these is. In biostimulation, you add nutrients and oxygens to contaminated areas, hoping to see an increased growth and activity of naturally occurring bacteria that will break down the contaminant. So you're adding the nutrients and the oxygen that these bacteria will need, hoping to encourage their growth and encourage their activity so that they can help to break down whatever contaminant it is that you're hoping to clean up. So that's biostimulation. So you're trying to stimulate these naturally occurring bacteria in the environment to do their job. Then we have bioaugmentation. So here you have organisms that can help to clean up a particular contaminant, and you add these to the area. So you're augmenting, or you're adding something. And this tends to work best when you have a contaminant that has been removed from the area and moved to a site where you can better control the conditions for the growth of that organism and really optimize those conditions so you can really have those organisms performing at their best. So that's bioaugmentation. It doesn't work so well on site, but it works best if it can be moved to a controlled environment. Then you have intrinsic bioremediation. And in this case, this is where this takes place naturally in water and soil. And sometimes you see this in contaminated sites where after a certain time, bacteria naturally begin to grow and break down the contaminants. So the bacteria that are carrying out the bioremediation are intrinsic to that environment. They're naturally occurring in that environment, and after a while, they really kick in and start decontaminating the sites. And the reason that these types of processes work is that you have different bacterial species that can use the contaminated molecules to release energy to power their own biological processes. So they use these contaminant molecules as their source of energy to fuel themselves. When that contaminant molecules gets broken down, energy is released. So scientists are studying different types of bacterial species to really try and determine which ones are best suited for which contaminants. For example, in gold mining operations, you have cyanide, which is a very, very poisonous material. Cyanide is used to remove gold and extract gold from the rocks in gold mines. And the problem with that is that then cyanide tainted water gets released back into the ecosystem, and that ends up killing many of the living things in that area. Now, some companies are using bacteria that are very good at breaking down cyanide to try to treat the water until a good deal of cyanide is removed, and then releasing that cleaned up water back into the ecosystem. So this has been tested. There's areas which have already been contaminated, and when these areas were exposed to the type of bacteria that consume cyanide, you could see that there was a decrease in the cyanide levels to the point where life started to thrive and that area started to grow again. There's also research taking place to try and use bacteria to clean up oil spills, both oil spills that happen on land and also the ones that take place in lakes and oceans. And it's the hope of scientists that the bacteria will be able to break down oil molecules and as a result help to clean up the environment, removing the oil molecules from the environment. And back in 1989, when Exxon Valdez ran aground and released, a 250,000 barrels of oil into the waters of the coast of Alaska, it contaminated about 300 miles of the coastline. And this disaster really provided the incentive that scientists needed to study how different bacterial species could be used to help clean up that region and from oil. There's also phytoremediation, and phytoremediation is a form of bioremediation, but instead of bacteria, it uses plants. And in this process, plants are used, and those plants' natural ability to absorb, contain, degrade, and remove these contaminants from water and soil. And scientists have been using these types of plants, such as sunflowers, such as ragweed, cabbage, and geraniums, to help to clean up contaminated sites. And there are many advantages and disadvantages to using this type of remediation. Plants are environmentally friendly, and oftentimes they're cost effective, they're not very expensive, and they have the ability to clean up a large number of contaminants. And in many cases, the metals that they absorb can then be removed from these plants and recycled. And because their root systems push down deep into the soil, they are able to prevent or reduce contaminants from entering the groundwater. Now, on the negative side, on the flip side, this type of remediation can take a long time because it relies on a plant's natural growth cycle. So the contaminants can only be removed from those areas during the plant's life cycle, and only as far as the plant's root system can reach. So this is limited approximately to about 3 to 6 feet for plants, and maybe a little bit more for trees, 10 to 15 feet for trees. But there's also a risk of animals, which will then be feeding on those contaminated plants while they are still on site, and then moving the poison into and further up into the food chain. And in phytoremediation, though, there are, again, different types of phytoremediation, and they are characterized based on whether that contaminant is a metal or an organic chemical. And if you have a metal that you're trying to extract from the environment, these methods of phytoextraction and rhizofiltration and phytostabilization are used. And if you have organic chemicals, there's these two methods. So let's look at them a little bit more closely. And excuse me for one moment. All right. So one of them is phytoextraction. So here in phytoextraction, the contaminant is taken up by the plant from the soil by the plant's root system. So here you can see that the contaminant that's in the soil can only be removed as far as the roots have access to soil. So it only cleans up the certain portion amount of soil that is accessible by the roots, and then gets taken up by the roots, gets taken up further into the plant tissue, into the shoots, into the leaves, and eventually this plant can then be pulled out of the soil and harvested, and with it, the contaminant can be removed from that portion of the soil. So that is phytoextraction as one way of removing contaminants, particularly metals from soil. Another method is called rhizofiltration, where the plant either absorbs the contaminant directly into its roots or is attracted to and held by the roots. And this only works for surface or groundwater remediation, and plants that are grown in clean water then get taken and are planted into the contaminated areas where the roots become saturated with the contaminants, and then they are removed and replaced with fresh plants. And this process has actually been used in artificially created wetlands to treat the wastewater and contaminated water from landfill sites. So this has been definitely used. And then another method is phytostabilization. So in this case, rather than absorbing contaminants, plants are used to contain or at least slow down the movement of these contaminants. And this can help to prevent the contaminants from getting into groundwater by helping to sequester them. And this picture is using phytostabilization, where the contaminant gets absorbed or just sticks onto the surface of the root and is just contained in the soil around the plant, stopping it from leaching further down into groundwater. So that's for metal contamination. Now, if you have organic chemicals, there are two additional types of soil remediation that can be used. One is called phytotransformation. The other one is rhizosphere bioremediation. In phytotransformation, plants are able to excuse me, absorb the chemicals and actually absorb them and break them down into less harmful molecules by metabolizing. So that's phytotransformation, where it gets transformed. The contaminant actually becomes transformed to a different type of molecule, a less harmful compound. In rhizosphere bioremediation, in this case, it's not the actual plants themselves that break down the contaminant, but instead they provide natural substances that then the microorganisms in the soil, such as yeast and fungi and bacteria, use in order to grow. And by providing these natural nutrients for these microorganisms, they stimulate the growth of these organisms, and as a result, use the metabolic processes within these microorganisms to break down the material that's in question. And so there's quite a bit of research into the different plant species that can be utilized in this way, and it's hoped that eventually a seed bank of plants can be developed that will be useful for different contaminants, so that in the future, if you have a site that is found to be contaminated with a certain contaminant, there will be a plant species that will be specific to help clean up that contaminant and solve the problem. And as a result, using plants is really the more environmentally friendly way than the complete removal of the contaminated soil and earth from the area. And it's also a good way of making use of the natural resources that are available to us. So this brings us to the end of our last lesson in Unit 2. So next week, Wednesday, or not next week, this week, next lesson on Wednesday, excuse me, I will introduce you to your project and presentation portion of this course that you have to do. I'll introduce you to that so that you can have plenty of time to think about what you might want to prepare for your presentation and what you would like to work on as your project. And I think we can also then start on our unit 3. All right. I will also post Unit 2's assignment, and I will post the due date for that. It won't be due this Sunday. I'll give you some time. I know this Sunday is a long weekend. There's Thanksgiving on Monday. So yeah, I think I will extend that deadline from Sunday to later on in the week. I'll let you know. All right, everybody. If there's no more questions, we can conclude our lesson here. Anybody have any questions? All right, everybody.
Good night, Miss.
Have a good night then.
Bye, Miss.
Bye-bye.
Thank you. Bye.
Goodbye.